2, we will start with talking about once you have cultures, how do you preserve them for future use? Okay, so you can prepare freezer vials called cryotubes of concentrated bacteria. So you take, maybe you get probably a 50 ml tube first before you get to the cryotube. You get a 50 ml centrifuge tube. You centrifuge down all the cells from your liquid media. Okay, that's going to make a pellet at the bottom of all the cells, and then you can pour out the broth, the growth media, and then you can resuspend that pellet in just a very little bit of whatever the suspension is that you want to freeze them in. Usually it is some media with some extra additives like DMSO to keep the cells intact when they freeze. So you would transfer that into the cryovial, which is usually only 1 ml. I think the biggest cryotubes I've seen have been for 2 mils. And of course, you wouldn't fill it all the way to the top. You'd want to leave space for it to expand when it freezes. And also, you never want to be touching the lids and risk contamination, right? Okay, so then you have your cryotubes, and what do you do with them? You can stick them in a deep freezer. Typically, this is a freezer that would be set at -80, but there can be a range. And we don't want to shock the cells, so we don't put them straight into the freezer at -80. We actually put them in a little container that has ethanol in it, and that will help kind of let it slowly freeze. Sometimes the additives that you put directly into it also do this, help it to slowly freeze, so you don't damage the cells. And then you can just leave them in the -80 for quite some time. I think we would allow a year for our stocks, and then we would need to make new ones. And once you're ready to use them, you just simply grab them out. You can let them defrost for a little bit at room temperature, or you can stick them in a water bath. And then you transfer the desired amount. Sometimes that's very small amount because it's super concentrated. It just depends on what the applications are. Okay, the other thing you can do is store them in liquid nitrogen, which is even colder, or you can freeze dry them. So liquid nitrogen is going to give you a little bit longer time, maybe up to 2 years. And then freeze drying is going to give you the longest amount of time. It also allows you to store those stocks either at room temperature or in a regular refrigerator. So it removes the need for these things, which can be quite expensive. Running a deep freezer, purchasing a deep freezer alone, that equipment is very expensive. And not everyone has access to liquid nitrogen. So another reason why someone might freeze dry. Plus, if you already know that that's going to be a long-term stock, it can be easier than trying to remember to make new stocks on a regular basis. In order to freeze dry, you go through the process of freezing your stock. Then you put them in this special vacuum, which is also keeps it frozen. So you set it to the temperature you want, and then the vacuum removes all of the water from the sample. And so this is commonly done when you're going to ship stocks. Now, there are also plenty of people who will pay the extra money to ship freezer liquid nitrogen in to keep the stocks, so you don't have to life size them, and that's fine. Just trying to introduce you to some of the different things that you might see if you end up working in a lab in the future. Okay. All right, so now, How do bacterial cells or prokaryotic cells replicate? And with an exception, remember that yeast are actually a type of fungi, and fungi are eukaryotic. So, this one branch of fungi, the yeast, behave actually like prokaryotes in that they're single cell organisms that replicate through a simple process called budding, which we'll see a little bit more about budding um, in your simulation. This week, you'll be looking at the yeast life cycle, and that's something else I wanted to mention. I'm going to show you bacterial life cycles, and you're going to need to note that for yeast, and in particular in the experiment that you're doing this week, it is different. So take screenshots and notice the difference between your yeast life cycle and the bacterial life cycle that I'm going to present in a moment. Okay, so binary fission is how most bacteria will replicate. They simply double all of their contents, including their DNA and all of their ribosomes and everything else that they have, the cytoplasmic material, they double everything. And then the membrane and the wall will start to grow in between. And so you have a double membrane and a double wall so that each cell can be its own separate cell. And so this is during this division, this is when the cell, we say, has doubled in size. It doubles in size and then it pinches off and becomes two separate cells. And then it repeats this over and over again as long as conditions are favorable. There's plenty of food and there's nothing too harmful going on. If resources become limited, then of course the process of cell division is going to slow way down so that they can focus on surviving, right? Okay, there's furthermore another group of bacteria, the filamentous bacteria, that they fragment for reproduction. And we might see this later towards the end of class. We'll see. But for now, I want you to focus on binary fission and budding. And budding is most common for yeast, although we do have a couple types of bacteria that will also do that. Okay. So in binary fission, as I mentioned, everything needs to double. So you have your cell wall, you have your plasma membrane, you have your DNA, and the DNA needs to replicate. Okay, the cell needs to elongate and prepare for this division. The cell wall and the plasma membrane will begin to constrict at this what we call the cleavage site, and these what we call cross walls form and completely separate. So basically you get a cell wall growing all the way across on one side and all the way across on the other side, with the membrane directly inside of the cell wall. So you get the formation of two separate cells. While that's happened, it's important to notice that the DNA has already moved into the two separate regions, right, that will become the two separate cells, so that one cell has a complete set and the other cell has a complete set of the two DNA copies. Okay, this DNA is, as we talked about before, is a long, or not long, a big circular piece of DNA. So it's one piece of DNA, it's circular. It is still two strands of DNA. Okay. And then the cells become separate and they can repeat the process over and over again. And so since they will repeat this over and over again, when I said as long as the environment is favorable, that would be during what we call the log phase of their growth cycle. But before, they need to sense the environment. They need to know, are there enough nutrients available? Is there anything harmful in the area? And if all the cues say go ahead, then they have to turn on all of those processes to replicate the DNA, to make sure there's enough ribosomes and cytoplasmic material to actually divide in half. So we call that the lag right? Because there's no real change in the overall number of bacteria in the sample during that time. And this is a different amount of time depending on the exact species, genus and species that you're looking at. So, for instance, E. coli that I worked with quite a bit, that I used specifically to then be a host for bacteriophage. So this particular type of E. coli that we called competent E. coli, it would have a lag phase of about 2 to 4 hours. And then we would see the log phase after that. Okay. So for each bacteria, it's different. And this isn't really, we don't have a good time frame at the bottom. Like this is 0 and this is 5. So this could be 2.5. So maybe this is about 2 hours of lag phase. So kind of similar to what I was saying with the E. coli. And then it enters this log phase of growth where each cell is dividing and then over and over and over again. And so you get this exponential growth, right? As 2 cells become 4, and then 4 cells become 8, and 8 cells become 16, right? And then this doubling each division leads to this exponential growth that you see. But eventually, all of that division, all of this rapid cell division is taking up lots of nutrients. It's producing lots of waste. And so eventually the cells are going to have to slow down and enter what we call a stationary phase, where you see no overall change. The amount of deaths equal the amount of new cells, and so it stays stationary. But see, compared to the lag phase, where it was also overall no growth or death, the population already grew to a very high number because of the log phase growth. Eventually, even staying at this very, very slow level of growth where new cells equal dead cells, eventually they still produce too much waste and eat up too much food, and they will eventually start to die. So this is given that you're in like a beaker of media where you're not going to have a continual flow of new food and getting rid of waste, right? Okay. And so then eventually you're going to have more cells die than new cells being produced, and that's the death phase, which is slow and may or may not get all the way down to having them all die, but you will see a drastic decrease in the number of bacteria. Okay, so now when you look at the growth curve for the yeast, you're going to see an extra, you're going to see the log phase divided into 2 segments. Okay, so pay attention to that for the yeast. And that's important because we're talking about the difference between when they're going through fermentation and when they're going through cell respiration. So you need to notice which is which, why, what kind of change are you seeing and why, okay? So this is just a visual representation of this doubling, right? So as each cell divides in half, and this is starting from 1 cell. Of course, you would never actually start with just 1 cell. You would already be starting with hundreds, if not thousands of cells in your sample. But even just starting with 1 cell, you can see that after just 5 rounds of division, you have a huge, right? You have this huge or exponential growth, okay? How long it takes for a division to occur also depends on the type of bacteria, but I like to think of it as being a little less or even a little bit more than an hour. So on average, I think of it as 1 hour. But it is different for each bacteria, and we can calculate that. And we can calculate that for that bacteria under the conditions that we're supplying, right? So it's not always the same for that exact type of bacteria if you are changing the conditions, the amount of food, the temperature, right? Is it in a shaking incubator or is it staying perfectly still? How much buffer, if any, did you provide it, right? All of these factors are environmental factors that are going to impact its growth curve. Okay, but keeping everything constant, you could then determine the rate at which the divisions occur, and we'll see that in just a second. So this is just continuing on. So in this slide, we saw just 5 divisions where one cell divides and you get 2 cells, and then 2 cells divide, you have 4. And the way to represent that, right, anything to the 0 power is 1. Anything to the first power is itself. So 2 to the first power is 2. 2 to the second power, right, and so on and so forth. So how do we look at the number of cells for each generation? You start with 2 to the 0, and then each division after that you increase your exponent by 1. Okay, and then of course if you were to look at the actual number of those cells, it's 1, 2, 4, 8, 16, 32, and then when 32 cells all divide in half, right, then 64, and you begin to have really large numbers, right? So here's where we left off with 5 being 32, and then what if you want to know what's in 10 generations, how many cells would you have at that point? And again, remember, this is starting with just 1 cell, which is way less than you would ever actually start with. And so generation 10 would be 2 to the 10. What does that look like in actual numbers? We're in the thousands, right? 1,024. And when you're at the 15th generation, 2 to the 15, 32,768. And these numbers become really large and really difficult. How would you graph something with a range from 1 to 1,000,000? And this range, 1 to 1,000,000, can happen much more quickly than 20 generations because, again, you wouldn't actually be starting with just 1 cell. And so what we're trying to demonstrate here is that instead of using the regular number of the cells, when we work with bacteria and we do graphs, we actually use the log of the number. It becomes much easier to put this in a graph and even just to say these numbers, right? It's much easier to say 3.01 than 1,024. It's much easier to plot 4.52 than it is to plot 32,768, right? And so on and so forth. So the log numbers are going to all look small, but they actually represent larger numbers, right? And so all of that to say, okay, so to plot these and actually see what's happening, it's much more useful to use the log scale. And now that you've accepted that idea, it will be easier to then work with this equation. Now, working with this equation, we have an exponent, right? The 2 to the nth generation. And to solve for that generation, we need to bring it back down from the exponent down to where we can actually solve for it and isolate it. And to do that, we would have to take the log of both sides of the equation. So we're going to see log again when we're working with these equations, okay? So let's think about this. In t time equals n naught starting time times 2 to the nth power, which is going to give you the amount of cells that you have at some time, okay? And so, as I said, n naught is the initial, right at the starting time, whenever you start your growth of your population, you would take a sample of that population and find out how many cells you are actually starting with. So the initial population number, okay, and then you let them grow for some amount of time and you take another sample to see how many you now have at that time. So population at time t, and you could take a sample, you could take a small sample, like even half a mil or less, and plate that or measure its turbidity even in a photospectrometer, and you can see the amount of bacteria over time change. So you could take these every hour or every couple hours, however you wish to do it, or you could simply just give it several hours and just have the two points. But you could make a growth. You can make a bacterial growth curve by taking samples over time, right? So you're letting big population grow, and every so often you take a little sample from it and find out how many you have, and you allow it to keep growing, right? So you can make a growth curve yourself by hand. And in fact, we normally do this when we're in person. You will have a bacterial growth curve lab next week where you get to see kind of in a different way. Now, in that lab, it's going to introduce you to a little bit more math than what I'm showing you here, but what I want you to know is what I'm showing you here in this lecture, and also what I'm going to have you do for the homework for lab. So you do not need to worry about all the extra stuff that they're going to show you in the Labster, right? They like to do a lot of math, and then what tends to happen is when you're just learning it, it's hard to know what to focus on or what it even means by the end. So focus on this lecture, and then focus on the lab homework. Okay, so the equation I gave you, NT, T representing the population at time T, whenever you decide you want to go from time 0 to maybe 5 hours, right? And then NT would be in 5 for the 5th hour of growth, right? In naught, the initial population number, so whatever you started with, how many cells did you start with when you started your experiment? And then 2 to the nth, we're going to solve for n. So we would know how many cells we had at the 5th hour, and we would know how many cells we had initially. We would plug in those numbers, and we would then solve for n. So to solve for n, instead of using actual numbers, I'm going to keep these variables in place so you can see what it looks like at the end, and then you could actually plug in those numbers directly into this new equation, this reformatted equation that has isolated n. Okay, so if you want to know how many generations happened, how often it actually divided in some amount of time, which the example I've been using is 5 hours. So if you want to know how many generations happened during this 5-hour time, does it divide every hour exactly, or is it a little bit quicker or a little bit slower? Okay, so to find that out, solve for n. So you take the log of both sides, right? And then you subtract log naught from this side, right? We want to isolate this n. So by taking the log of both sides, you've now brought the n from the exponent down in front of the log. And now we want to get this n by itself. So we move this guy, log of n naught, over to the left by subtracting it from both sides. And then we're going to divide both sides by log of 2 so that we can isolate this n. So dividing both sides by log 2, you now have this. N equals the log of the time period, nt, subtracted by the log of n naught, and all of that divided by log 2. Well, log 2 that's an actual number that we can solve. We can put log of 2 in our calculator, and that gives you 0.301. So then now you have this nicer equation, n equals log of nt minus log of n naught divided by 0.301. So you should memorize this starting equation, nt equals n naught times 2 to the nth power, and then you should either be very, very comfortable with solving for n and be able to do this process without making any mistakes, Or you can memorize what the answer is. N equals log nt minus log n naught divided by 0.30. And you know, once you have the number for your ending, right, the concentration of cells you have at whatever time period you choose, subtract the amount of cells that you started with, and then you divide, right? Order of operations. So you just have to remember a little bit of your math that you guys have learned, some of you a long time ago, some of you more recently, okay? So when you grow a culture, you will measure the concentration of bacterial cells you have at your starting time and then at your end time or at each interval, and then you choose that and solve for that to see what your generation time. And then if you're taking them every hour for several hours, like half a day or more, you could get the actual curve. You'll be able to see as it's gone through the lag, the log, the stationary, and then even maybe start to see it die off if you go, if you're taking samples long enough. If you only take 2 samples, you may only see one part of its growth curve. So, and that just depends on the amount of time that has happened, right? So this tells us the amount of generations that occur roughly, right? It's almost like an average, unless you're taking times regularly. That would be the only way to get the actual growth curve, right? If you only had 2 points, this point here and this point here, it would just look like a straight line, right? You wouldn't know how long it spent in its lag phase and how long it spent in its log phase. You wouldn't even know if it's actually entered its stationary phase or not, unless you're taking samples regularly over time. Okay. Your book explains this very well, so I really suggest that you also read this entire chapter in your book to really make sure that you understand this, because this tends to be another area that's difficult for people. Okay. So this is what we ended up with on this slide, right? So the number of generations, n, little n, is equal to the log number of cells at time t minus the log number of cells that you started with initially divided by 0.301. Okay, so this looks a little nicer than how I wrote it here. It's hard to do type math. It takes a really long time to type it up nicely like I did over here. Okay, now this tells you how many generations happened during that time period. Okay, if you want to know how many generations actually occur every hour, then you simply put generation time will equal, and this is what you're going to solve for, 60 minutes times however many hours lapsed. So again, the example I've been using is 5 hours. Then you would put 5 hours times 60 minutes, gives you overall how many minutes, and then you're going to divide that by n, the number that you have here. So number of generations is little n, right here is little n. So how many generations actually happened during that time period divided by the total amount of minutes that elapsed, and that gives you how many minutes per generation time. Okay, so here I put minutes per generation, and here I put generation time, and that's really the same thing. Okay, so why don't you actually practice plugging some actual numbers in? If you start with 100 cells in a sample and you let them grow for 5 hours, and at the end of the 5 hours you see how many cells you have, and you actually have 1,720,320, right? 1,720,320 cells. Blah. Okay. Now, solve for how long the generation is. In order to solve for how long the generation time is, you need to first do this first equation, number of generations during 5 hours, and then use the second equation to find out how long each generation is. Okay, pause the video and actually solve this question. All right, did you pause it? Here's the answer. Check your work. So you should have plugged in At the end of that 5-hour period, this is how many cells you had in your sample. You're going to take the log of that number. You're going to then subtract the log of 100, how many you started with, and then divide by log 2 or divide by 0.301. Okay, so you do that and you find out that 14.072 generations happened during that 5-hour block of time. It divided just a little bit more than 14 times. So that's actually really fast. Now, how fast is that exactly? 60 minutes times 5 hours divided by your number of generations, little n, and that gives you 21.32 minutes. So every 21.32 minutes, it's dividing in half. Whew, that is fast. I made these numbers up. Okay, I don't think we have any bacteria that's quite that fast, but perhaps because, remember, they go through a period of log phase that's super fast, and then the other growth is slower. So, it's possible. Anyways, the point here is that you know how to use the equations. You can make up some more numbers. You can make up a starting concentration and an ending concentration, and then you could practice these equations again. And actually, something that would be fun is if I gave you some data for maybe, let's say, 6 time points, and have you actually make a growth curve like we would do in lab. So maybe I'll add that to our module as an optional practice, or maybe as an actual assignment. We'll see. I'll let you know. And I'll always give you plenty of time to complete things, so don't stress. Okay, so now I want to talk about biofilms, kind of take a little break from math, think about biofilms for a little bit, and then we'll switch our gears back into doing a little bit of math. All right, so biofilms, we've talked about these before, right? They can form on your teeth, they can form on water filters, they can form on medical equipment, right? And they can be very problematic for patients and having basically recurrent infections that are not treatable with antibiotics, right? Because you're never actually getting rid of the actual source, the biofilm itself. All right, so biofilms are a kind of complex microbial community, and it doesn't even have to be just one species. It can actually be mixed, but we tend to show examples of single, okay? And remember, they're forming this special kind of like slime or hydrogel, thick, film, biofilm. And so bacteria, as these slimes or hydrogels start to be secreted by bacteria, more bacteria are going to come. They're going to be attracted to it. And so this begins this idea of quorum sensing, okay? So more bacteria join in, and they start to excrete this substance, and they'll all keep doing this until they sense that there's enough, okay? So quorum sensing is a little slightly, advanced concept because there's a lot of different applications. Quorum sensing doesn't only apply to biofilms, and the exact way in which it occurs can vary, but I just want you to get a general idea for it, okay? Now, not all bacteria are capable of forming biofilms, only some species, but it's not only bacteria. There are some types of fungi that can do it too. But when they're in this community, and let me just show you what it looks like, you see there's like groups of them, and they're going to be sharing nutrients, and then their waste will be taken away because of these column-like structures that they're forming. And again, this is at the microscopic level that we're looking at, so it wouldn't look like there's all these gaps when you're just looking at a biofilm. It just looks like a solid film, right? But what's actually happening is that there are these little pores throughout that allow nutrients to come in and waste to be swept away. Okay. Little clumps can break off and allow the film to travel and find a new area. And furthermore, sometimes the bacteria will break down their film, regain their flagellum, swim around, and then can start this process again as well. So they are a community, as you saw. They're sharing nutrients and they're sheltered from harmful factors. The bacteria that are inside here, they're safe. Nothing you throw at it, no chemical is going to kill the bacteria that are in the inside of the biofilm. Okay, so I want to go over the steps of biofilm formation. I've written them out here. These are the steps I want you to know. But instead of me just telling you word for word what's happening, I want to show you. So we're going to watch this video, okay? So let's take a look at it. We have, I just need to resize
bacteria are
so that you can actually see it. So let's see. It's tricky, isn't it? So you can see that whole big space. Okay. So let's make this a little bit. Okay. So I can't really optimize the size of this video for the part of the screen that you can see, but you have the link, so you can watch this again in a bigger view. But for now, you'll at least get the general idea.
Hi, my name's Bonnie Bass.
Oops, looks like I accidentally clicked on a different video. Here we go. That's also not it. It's so odd. I don't know. I guess that's what happens when I try to move things around. Okay, here we go. Biofilm formation.
Bacteria are typically thought of as unicellular, but in nature, many, if not most bacteria, form specialized surface-attached communities called biofilms. This micrograph depicts a biofilm of bacteria collected from a rock surface in a Yellowstone hot spring. Even in aquatic environments, bacteria are found mainly associated with surfaces. Plaque is a biofilm that forms on teeth. Biofilms play critical roles in microbial pathogenesis and environmental quality, and cost the nation billions of dollars each year in equipment damage, product contamination, and medical infections. Most biofilms in nature are consortia of several species, but in some situations, single-species biofilms can form. For example, the gram-negative bacterium Pseudomonas aeruginosa can form a single-species biofilm in the lungs of cystic fibrosis patients. Single-species biofilms are also readily formed in the laboratory. The first stage of biofilm formation occurs when a specific environmental signal induces a genetic program in free-living planktonic cells. In response to the signal, the planktonic cells begin to attach to nearby surfaces by means of flagella, pili, lipopolysaccharides, or other cell surface appendages. The attached cells begin to coat that surface with an organic monolayer of polysaccharides or glycoproteins, to which more planktonic cells can attach. As the cells enter into biofilm formation, they no longer maintain their flagella, and instead may move along surfaces using a twitching motility that involves the extension and retraction of a specific type of pilus. Ultimately, they stop moving and firmly attach to the surface. As more and more cells bind to the surface and divide, they form microcolonies and can begin to communicate with each other by sending and receiving chemical signals in a process called quorum sensing. These chemical signal molecules are continually made and secreted by individual cells. Once the population reaches a certain number, analogous to an organizational quorum, the chemical signal reaches a specific concentration that the cells can sense. This concentration triggers genetically regulated changes that cause cells to bind tenaciously to the substrate and to each other. Quorum sensing may influence the amount of extracellular matrix produced by the cells. This thick, slimy material consists of polysaccharide polymers called exopolysaccharides and entrapped organic and inorganic materials. The extracellular matrix protects the cells from damage from environmental assaults. Quorum sensing serves the biofilm in many ways, such as by increasing the resistance of biofilms to antibiotics. Quorum sensing in Pseudomonas aeruginosa plays a role in the maturation of the biofilm into complex three-dimensional forms, such as columns and streamers, forming channels through which nutrients flow. Sessile cells in a biofilm chemically talk to each other in order to build microcolonies and keep water channels open. Biofilms tend to form where nutrients are plentiful and may begin to dissolve when nutrients become limiting. Little is known about how a biofilm dissolves, although the process is thought to be triggered by starvation. Pseudomonas aeruginosa produces an enzyme that can strip away the exopolysaccharides. Cells leaving the biofilm revert to their flagellated planktonic forms and can swim to new locations where nutrients are more plentiful. Pseudomonas aeruginosa can be medically devastating. It forms biofilms in susceptible individuals, such as in the lungs of patients with cystic fibrosis, and on medical implants.
Okay, so you can, I highly recommend watch that a few times. You can also turn on the subtitles for that video to help you get all that information. And as I mentioned, I already wrote down the essential steps that you must memorize. So an environmental signal induces the genetic programming in bacteria that we call planktonic. This means they're free, they're swimming, right? They have their flagella. And this signal tells them to start to adhere to the surface, to a surface, and start slowly to start secreting some of those polysaccharides in the biofilm, which will then lead to a more firm attachment onto that surface. It'll lead to losing the flagella, right? And it's going to signal more cells to do the same. And as you get more cells together on that surface, close together, doing this process, eventually they produce so many of these chemicals that talk to each other that they then really increase the rate of biofilm formation and form the actual biofilm, right? So this extracellular matrix forms around them and forms the actual official biofilm, where you then get the 3D structures with those columns and streamers forming these microcolonies, which allow the water to flow through so nutrients gets in, waste is removed, and they can sustain those biofilm colonies for quite some time until food runs out, right? And then they can degrade the film and leave, revert back to planktonic, right, flagellated cells once again. And once they find another surface with the right environment, right, good nutrients, they can repeat this. Okay, so that's biofilm formation. Okay, make sure you can answer the check your understanding questions that you see at the end of each section, right? Okay, so now we're going to look at how do we count, how do we measure how many cells we actually have in a sample? So I showed you the equation for getting how many generations happen in a certain amount of time and how long is each generation. And I told you have to measure how many cells you start with and how many cells you end with at that point in time. And so now we'll talk about how do you actually do that? How do you get those numbers, how many bacteria you have in your sample? And there's a few ways. There's plate counts, which is the predominant, is one of the predominant ways that we do this. And there's also a lot of different applications for using plate counts, not just to plug in to the generation equations that we saw, but also just to understand how much bacteria are in a sample, to know like how contaminated is some water source, for example, or how many bacteria are in this person's mouth, or whatever. How many bacteria can this disinfectant kill? How many do you start with? How many do you end with? Right? So plate counts, right? So after you have your sample, you will plate some amount of that sample onto an agar plate. You will then incubate it, and that incubation could be 24 hours. It could be 48. If you are interested in a specific type of bacteria that's slower growing, you might give it even longer incubation time. So that all those, how much you plate and how long you incubate it, that all depends on your specific application. And then after the incubation, you're going to count how many colonies are on the plate. And because typically you have a lot of bacteria in a sample, in order to actually get colonies that you can count, you need to do a series of dilutions and plate each dilution tube, because you don't know which one's going to produce the right amount that you can count. And so it's a series, okay? Now remember, we talked about colonies before, and they represent a single bacterium, and so that's bad grammar on my behalf. So it should say bacterium, IUM, because you're talking about one individual bacterial cell. So one bacterium, it lands on that spot on the agar, and it divides over and over and over and over again to form a colony. Okay, we call that a CFU, colony forming unit. I don't know why we can't just call it bacteria, but this is how it works, okay? So we call the amount of colonies on a plate the amount of CFUs. So you would say, I counted 25 colonies on this plate, so there are 25 CFUs in the sample that I plated, okay? Okay, so it's tricky when colonies are touching one another to know exactly how many colonies are there in that spot. So that's another reason why we do these dilutions and plate everything, so that we will actually be able to have plates that we can use and count. So if one plate has tons and tons of colonies on it, and they're all touching each other and it's too difficult to count, we call it TNTC, too numerous to count. It would not be accurate to try to count those. Even if I know a lot of people are saying, well, if you just divide it into quadrants, you count one quadrant and then you multiply it to represent the total population. But that only works when you can actually count the colonies in that quadrant. Okay, so here's an example. Here's a series. We have some sample here, and we do a dilution. And this is a serial dilution because you're doing the exact same thing over and over again. So we have a serial dilution that's going to be used to make plates and count them, and then we can do a simple calculation to find out how many we actually had in the sample. And you can see you're not even getting individual colonies. It's so concentrated, it looks almost like just a solid growth. We would not be able to count this. We would call this TNTC. This one's getting better, but this still has regions of very, very solid growth, and we would not be able to distinguish how many colonies are on this plate. So we would also call this plate TNTC. But now, after we've diluted it again, now we see that we do have individual colonies that we can count. Yes, a couple of them are still touching, but you can clearly see that this is 2 colonies touching, right? And you have to use your judgment, and then you have to stay consistent in whatever you have decided on how to count those few colonies that do end up touching. And you would count this plate and you would count this plate. However, this one, even though it's obvious and it only has 3, typically that is not enough for us to feel confident. Now, if you were to do the calculation and it gives you the same exponent of how many bacteria are present, and so it's in agreement with the other 2, then you can use it. But if it gives you a different exponential number, which tends to be the case, we don't include it because it's too few. Furthermore, the actual cutoff on how many colonies are too many and how many colonies are too few would be determined by the size of your plate. And so you would know that when you're doing your experiment. You would have an actual cutoff number, like anything less than 10 is too few for a large plate. But for a small plate, it might be anything less than 2, right? So it just depends on the size of the plate. All right, so now that we understand the overall, let's go ahead and examine one in particular so that we can actually count how many bacteria or CFUs we had in our sample. So you can count how many colonies are on your plate, and this would be the amount of CFUs. You can count what your dilution factor was to make that plate, and you also need to know how many milliliters were plated, okay? So the information you need to know is your total amount of CFUs on your plate, how many colonies you have, how much of the sample, how much of the dilution tube was plated, and what is the dilution factor of that tube, okay? And then you're going to plug it in. So you see here colonies on the plate, CFU, divided by the amount plated, in this case 1 mil. And then once you do that, you times it by 1 over your dilution factor. So your dilution factors are going to be negative exponents, right? So we diluted this 1 to 10, right? 1 mil into 9 mils gives you a total of 10 mils, so 1 to 10. And so that gives you 10 to the negative 1 as your dilution factor. And then you put 1 over 10 to the negative 1, and that makes your number now positive. Basically, it takes your exponent and makes it positive, right? So instead of 1 over 10 to the negative 1, I simply just write 10 to the 1 because that's going to be the same. So whatever math you are more comfortable with, you can use. But you will see me doing the just simply getting rid of the negative on my exponent and not using the fraction because it'll mean the same exact thing mathematically. Okay, so each dilution in that series that we just saw, it was a serial dilution set, had the same dilution factor of 1 to 10. And remember, 1 to 10 can be written as 10 to the negative 1. And so you could go back to that image and you could label each of these dilution factors as 1 to 10 to 10 to the negative 1. And then you could also say, okay, well, what's the total dilution factor, right? So instead of just from this tube to this tube and this tube to this tube, right, we know from in between each tube is a 1 to 10 dilution. We also want to know the total dilution factor in order to use it in this equation. What's the total dilution factor for this second tube? And so that would be this times, right, each dilution factor times itself. So we had a 1 to 10 dilution factor in this tube, and we had another 1 to 10 dilution in this tube, you multiply those together, and that gives you 10 to the negative 2. And you do that over and over again for each tube, okay? So, in order to calculate the amount of bacteria per milliliter in your sample, and remember, we represent the amount of bacteria in the sample by CFUs. So what are the CFUs per milliliter of the sample? You need to know how many CFU were on your plate, how many mils you plated, and then times it by 1 over the dilution factor. Or as I said, instead of using this whole writing all that out, instead of putting like 1 over 10 to the negative 1, I just simply write 10 to the 1, right? So, sorry, this is sloppy writing with the mouse on my computer, but if you were to put if you were to put 1 over 10 to the negative 1 in your calculator, that's going to equal 10 to the 1. It's going to get rid of that negative. Okay, so I do that in my head, and I just write 10 to the 1. If that's my dilution factor was 10 to the negative 1, then I know that I'm going to use 10 to the 1 here in this equation. So that's what you'll see in the rest of these examples. Okay, so then let's say we want to look at that third tube in our example here. Let's say we want to look at this guy, because this is the first tube that's giving us countable colonies. That's why I selected this one. It's the first one that gave us a countable number. Okay, so we know that each tube has a 1 to 10 dilution. Okay, so this is 10 to the negative 1. This is going to be 10 to the negative 2. This dilution factor is going to be 10 to the negative 3. And another way to write that is by saying the dilution factor of A times the dilution factor of B times the dilution factor of C, right? ABC just is what I'm calling tube 1, 2, and 3, right? So you could call this tube A, you could call this tube B, and you can call this tube C. And then you multiply each of their individual dilution factors times each other to get the total dilution factor. Now, since this is a serial dilution and each one was 1 to 10, I was able to just do that automatically in my head, right? 10 to negative 1, 10 to negative 2, 10 to negative 3. But when you start working with dilutions that are not serial, or you just want to be extra careful, you can always do the A times B times C. Okay, so if you wanted to know the second tube, it'd just be A times B. If you wanted to know the fourth tube, it'd be A times B times C times D, right? So however many tubes to get to the tube of interest. In this scenario, I asked about the third one, so that's why it's ABC. Okay, so that gave us 10 to the negative 1 times 10 to the negative 1 times 10 to the negative 1 gave us 10 to the negative 3. And let's say instead of plating 1 mil, let's change it up and say that we plated only 0.1 mil, very small amount. And furthermore, let's say, and this is it, I don't know why there's a big space here, but let's say from this tube, we plated 0.1 mils, and after incubation, it grew 32 CFUs. Then we can plug all this information into our equation, right? So we have 32 CFUs divided by, and if we're going to change it up and plate 0.1 mils, then we would need to change this from 1 mil to 0.1 mil, right? And we would need to change that. So then we would actually be looking at 10 to the 5th power. So sorry for that mistake. I probably went back and wanted to make it a little more interesting of an example. So let me go ahead and erase some of this nonsense and make this a little more easy to read. So because I already have written it for 1 mil, I'll just change this. Right, we'll just cross this out and we'll just leave it as 1 mil. Okay, so let's say we plated 1 whole mil, just like it showed in the picture. We are looking at the third tube, so 10 to the negative 3 power. And let's say that plate had 32 colonies on it. So we have 32 CFUs divided by 1 mil times 10 to the third power. And that gives us 3.2 times 10 to the fourth CFU per mil. You have to be using scientific notation. You have to get comfortable with exponents. This here can be done without a calculator. If you're comfortable with exponents, you know the decimal point is here right now, and we always need to have it a single digit for scientific notation. So this is going to move over 1. Moving that over 1 is going to increase our exponent now to 4. We're dividing by 1, so that doesn't change anything. So then we get 3.2 times 10 to the fourth power. Okay, if this was 0.1, then we would be increasing it by another exponent. Okay, I will have the scientific calculator available to you on your test, so you can use the calculator. You don't have to worry about doing math in your head, but I do always like to show you just in case. And like I want things to make sense. So again, please correct this slide. You can either keep the 0.1 and change it here, or you can keep this and change it here. If you keep the 0.1, then you would need to increase this exponent to 5, right? And if you keep it as is, you just need to change this to 1 mil. Okay, thank you. Now, the next thing I want to answer is, what if we looked at tube D, the next tube? So we would need to multiply A times B times C times D, and that would give us a dilution factor of 10 to the negative fourth. Let's say we plated 0.1 mil and it grew 9 CFUs. So 9 CFUs divided by 0.1 mil times 10 to the fourth will give us 9.0 times 10 to the fifth CFU per mil. Okay, so do some practice, plug in the numbers, get comfortable. I gave you a big practice set in the module, just called, it might be called quantification of bacteria, or I can't remember what I called it, but it'll be right underneath this lecture slides for that practice set, okay? So right now, why don't you go ahead and practice while you have me with you, okay? So you do it. If you have 100 mil sample, so you're starting with 100 mils, and you do serial dilutions of 1 to 10, each dilution tube will have a total volume of 10, so you're putting 1 mil into 9 mils, and that gives you a total of 10, and then you plate 1 whole mil from those tubes. And let's say we're interested in just the second tube in the dilution set. After you plated 1 mil, you incubated it, and it grew 119 CFUs. So tell me, how many CFUs per mil were in that tube? This is, we didn't look at this. We looked at going all the way to the original sample, but I want to see if you understand what we were doing. So can you do it just to the tube itself? And then can you do the original sample, so the whole thing that we did together, and then take it a step further, and instead of looking at CFUs per mil, can you tell me just the total CFUs in the original sample? Okay, so now I'm asking you to apply what we did together. Do the first step, do the whole thing that we did, and then go a step further. Okay, did you attempt to answer these questions? All right, so if you have a 100 mil sample, you prepared 1 to 10 serial dilutions, and the second dilution tube, you plated 1 mil and it grew 119 colonies, CFUs, right? How much was in the tube how much was in the original sample and what is the total concentration of the original sample. Okay, so first, if you just want to know the amount of bacteria in that dilution tube itself, then you know that you plated 1 ml, it grew 119 colonies, and the volume was 10 mils. So there was 1.19 times 10 to the third CFU per mil in that tube. But if you want to know how much was in the original sample, you would take the CFUs per mil plated times the dilution factor, and that would give you 1.19 times 10 to the fourth power in that original sample. Okay, now the next question was total concentration. So instead of looking at CFUs per mil, we just want to know the total CFUs in that sample. Then you multiply the total volume of the sample times that concentration per mil that we just found in the previous question. And so then that's going to give you that container of the sample, that 100 mil sample, contains 1.9 times 10 to the 6th CFU, right? 1.19 times 10 to the 4th CFUs per mil. And if you times that by 100 mils, the mils cancel out and you get just CFUs. Okay, so there's approximately 1.19 times 10 to the 6th power bacterial cells in that sample. But instead of saying bacterial cells, we call them colony forming units. All right, how did you do? Did you do it? All right. A practice question for you. So go ahead and pause the video and solve this question. Okay, did you do it? Did you pause the video and solve for each of these questions? Okay, I'm going to trust that you did it and show you the answers now. So for the first question, it said, just show me the dilution factor for each tube. So tube A, you have, right, so this is not a serial dilution anymore. This is just a dilution series. So in this first one, you have 5 mils and you're putting it into 45 mils. So this is a big tube. So you're putting 5 mils and the total volume is 50, right? Once you add the 5 mils to the 45, the total volume is 50. So your dilution is a 1 to 10 dilution. So even though it's not 1 mil into 9 mils, you still got a total of 1 to 10 and have a 10 to the negative 1 dilution factor. In tube B, you put 1 mil in a total of 10 mils like you're used to seeing. And so that's also a 1 to 10 dilution, 10 to negative 1. And so tube B is A times B dilution factors is 10 to the negative 2. In tube C, we saw 0.3 mils divided by 3 mils total volume. I should have been writing mils on here. And that gives you 10 to the negative 1. And so if you multiply A times B times C, each of them being 10 to negative 1, that gives you 10 to the negative 3. So hopefully this is exactly what you had. Number 2, it asked you to find the concentration of the original sample if you use the third dilution tube, and that third dilution tube produced 8 colonies, and you plated 1 mil. So 8 colonies, CFUs, divided by 1 mil plated times your dilution factor, right, 1 over your dilution factor gives you 10 to the 3, and that gives you a total of 8 times 10 to the 3 CFUs per mil. So that's your original concentration of your original sample per mil. If you're asked for total concentration, then you multiply by the volume of that sample, and then that gives you the actual total concentration, so just CFUs. Okay, good job. There are 2 more questions I asked. I asked, what about if we looked at a different plate? What if we had a plate with 79 CFUs, but we had plated 10 mils, the whole tube? And would that agree with what we just did with this sample here of just plating 1 mil? And so you do the math, 79 CFU now divided by 10 whole mils times the dilution factor, still looking at that third tube, so 10 to the 3. So now you get 7.9 times 10 to the 3 CFUs per mil. What did we have before? We had 8 times 10 to the third power. That is almost exactly the same number. This is great support that whether we plate 1 mil or whether we plate 10 whole mils, we still get the same original concentrations, the same power, 10 to the 3. So that is great support. However, that's only 2 plates, right? So we've confirmed it once. The gold standard for microbiology is to do things in triplicate. So to have 1 more plate, you would have not only 1 confirmation, but you would have even a second confirmation. And then we would feel pretty confident that it's correct. Okay? So that's what we mean by triplicate. Having the 3 plates with colonies that are all giving you a concentration to the same power, 10 to the third power. Okay? In this case, 10 to the third. With other examples, it could be different, but they should all have the same exponent when you get to those concentrations. So the concentration of the sample is CFU per mil. The total concentration would just be CFUs. So make sure that you're really careful with your units, okay? All right, so for more practice, go to that practice set. In office hours, once you've gone through doing those practice questions, then we can go over the answers, okay? So I'm not going to go over answers this week. I want people to have time to do it, but the following week, we could go through them. Okay, so what about samples that don't have very many bacteria in them? Instead of doing dilutions, you need to actually do the opposite. You need to concentrate it. So you can run your sample through a filter and collect all the microbes in that volume that you have and grow that on. So you wouldn't see this because this is an electron micrograph here, but these are the pores of the filter, and the bacteria don't fit through, so they get trapped on the filter membrane, and then you put that membrane on the plate, and then you incubate it, and then after the incubation time, you'll see the colonies growing on the filter plate. And then you would have to do a calculation to find out how many bacteria you actually had per mil. We're not going to worry about doing the math for this. I just want you to know the concept, right? So when the total amount of bacteria in a sample is presumably too low to be able to plate directly, we do filtration to concentrate it so that we can get enough colonies to count. And again, we're not going to do the math for that for the filtration, but I want to introduce you to this idea. Okay, another way to count how many bacteria you have in a sample is to actually directly count it under the microscope. You can take a sample of your bacteria in your solution, your population, and use a specialized plate that has a grid on it. And so there are different kinds, and it would just depend on the lab that you're in. And depending on the cell counting slide that you use, you would know the area and the depth of that slide. So you know how much volume of the sample you're actually looking at. And then you will see these grids, and you'll see dark, thicker grids, with smaller squares inside. And if this is your whole field of view, then you might only count this one square, right? Because I don't know how many bacteria total are in these other squares because I can't see them all the way, but I can see this one all the way. So maybe I just only count the bacteria in this square, and I learn based on what the manufacturer tells me of what the volume of fluid is just in this one square, and then I do the math and I find out what that means, how many bacteria there are per mil of my sample. So this is called the direct microscopic count. I'm also not going to ask you to do this math because I really want to focus on the few things first. Master the plate count, master the generation time, right? And then when you get into a lab and you need to do these things, it'll be easy for you to learn how to do them because you already have a strong foundation in the concepts, okay? Typically, however, you would be able to see more than one square at a time. You could just reduce your zoom, right? Go to a little lower power lens on your microscope so that you could see the entire squares on the sides, and then you would count maybe 1, 2, 3, 4 of those big squares, or even 5, and then average them out, and that gives you a more accurate number, right? Because maybe there's a lot in this square, but maybe there's very few in the other square. So the more of these that you count, the more confident you can be in your total concentration. Okay. All right. So I did write out some instructions here for the steps that you would do just to emphasize the concept. So in your textbook, when you look at this, it's going to give you different information. I retyped the instructions here to make it more general, just to get you the concept. Okay, so make sure you read this slide rather than your textbook. You can look at your textbook to make sure you understand the concept, but no, you notice that I did actually change it to make it easier so that it's focusing on the concept instead of giving you specific numbers. Okay. The other thing, which I mentioned earlier, is you could take a sample and you can look at its turbidity. How cloudy has the sample become? So the blank would be the growth media without any bacteria. So we know how much light passes through growth media without any bacteria, and that would equal 0. So this would give us our starting point. Then we have our sample that has bacteria in it, and we measure how much light can pass through it, and then we have something to compare to, right? And you can use this as a rough estimate of how many bacteria are in your sample based on how much light is able to pass through it. And this becomes better the more samples you have, the more comparisons you have. And so we would say we want to know the absorbency, or we want to know the transmission. So you would need to specify, are you looking at how much light is absorbed by the sample, or are you looking at how much light is transferred through the sample? And then, so you could play absorbency or transmission, but usually we look at absorbency, A, okay? How much did it absorb? And that gives you an idea of how turbid the solution is. So this is a very common but indirect method. We're not actually counting bacteria directly anymore, right? We're not looking at colonies, and we're not looking at individual bacteria. We're looking at just turbidity as an estimate of how many are there. Okay, so this is an indirect method. There are more methods that can be used to count your microbes in a sample. Direct methods that we didn't talk about would be the most probable number. It's explained in your textbook. You can definitely look at that explanation for a much better idea. But to give you a rough idea of what the most probable number is, it's when bacteria don't grow very well on Petri plates, solid media, And so you want to use growth media in tubes, and you do a set of dilution tubes, and then you do some little statistics using those dilution tube sets. Your book has a very nice image and explanation of that, how that works. And this is pretty common in like wastewater treatment plants, for example, where you're working with bacteria that are not used to growing on a solid petri plate, right, that don't grow well. There's other applications as well, but that's still a direct method. It's still considered a direct method, okay? For indirect methods, we have the turbidity that we just looked at using that photospectrometer to measure absorbency. We could also look at the metabolic activity, and that really depends on your application, but typically for a metabolic activity measure, you're measuring how much CO2 gas is being produced by the organism over some amount of time. And that's used a lot with like mice, for example. If we were doing an animal study, it's real easy to do that with mice. It's used for different applications, not only mice. Dry weight is very common when you have filamentous bacteria or filamentous fungi. And so then you would dehydrate the sample and then take a measure, and that would give you the dry weight of the organism, and that would then tell you how much you have. So this is your overview of measuring microbial growth. How many organisms are in your population. Okay, that's it. That's the end of the chapter. So take your time, practice the math, practice these concepts, and read your chapter. Your chapter is very, very useful. Okay, good job.